Okay.
Okay, so here we have some histological images of other, of the lymph node and some other secondary lymphoid organs. So let's talk about how you'd tell them apart. Okay, so we've got the lymph node here. The lymph node, remember, is going to have the cortex and the medulla. Okay, so when you're looking at a specimen of the lymph node, you should see the cortex with its denser arrangement of tissue and also with those nodules in it that have the germinal centers in the middle. Okay, and then if we compare that to the medulla, and they've got a little bit of the medulla over here, you can see how the density of the tissue becomes much lower, and you have these nice channels running through there, what they call the medullary sinuses. Okay, so this is one of the places, remember how we talked about that lymph coming in and hanging around for a while. This is one of those places where it would be hanging around while it's waiting to get out into the efferent vessels and away from the lymph node. Okay, so cortex, denser, superficial, medulla, deeper, looser arrangement, kind of strand-like arrangement of cells with the spaces in between, the sinuses in between. Okay, so those are some histological clues for how to identify the lymph node. Over here we have the spleen. Okay, the spleen, if you remember from your notes from lecture, has two types of tissue within it. The red pulp, which makes up most of the tissue in the node, and the white pulp. Okay, the red pulp is the part of the spleen that does the blood-associated functions. Okay, so when you talk about the spleen as being the place where you have your red blood cell graveyard, that's the red pulp that you're talking about. Okay, the white pulp is the immune portion of the spleen. And the white pulp is always concentrated around branches of the splenic artery. Okay, so when you look at the white pulp, and it won't be white in the specimen because of the stain that they use. It's usually darker stained than the surrounding tissue, just as it is on this image. See how this white pulp is kind of darker blue than the remaining tissue? Okay, and in our specimen in the lab, the white pulp is purplish, kind of a darker purple, and the red pulp is more of a lighter pinkish purple. Okay, but you'll clearly see when you look at the specimen in the lab that there's a little branch of the splenic artery in the middle of the white pulp. Okay, so look for the artery and the darker tissue around it, and that will be a good indicator for the spleen. Okay, we'll move down to C here, the tonsil. Okay, and the definitive histological feature of the tonsil is the crypt. Okay, so all tonsils have deep indentations within them that are connected to the oral cavity. You can see one here that's connected to the surface. You can see several others over here that have their connection in some other location. Okay, but if you look in and you see tissue and you can see that there's a definite elongated space within it, that's got to be the tonsil with the crypt. Okay, and many times you can also see the stratified squamous epithelium along the edges of that crypt as well, since that's continuous with the epithelium of the oral cavity. Then we have Peyer's patches, and Peyer's patches are found in the wall of the small intestine. So we're kind of jumping ahead a little bit for morphology of the small intestine, but the small intestine is a digestive organ, so it's basically shaped like a hollow tube. So in the center of the tube you're going to have the villi that have a finger-like structure, and then you'll have a layer of connective tissue where the Peyer's patches are found, and those are just a set of lymphoid nodules kind of packed together into one spot. And then as you move toward the external surface of the organ, you're going to see a couple of layers of smooth muscle. And we're lucky in the specimen that we have that the smooth muscle is very obvious and distinctive. Okay, so look for the fingers facing the lumen, the smooth muscle closer to the wall, and the lymphoid nodules in between. And that's a good set of clues to put together for the Peyer's patches. Okay, so all of these secondary lymphoid organs are going to have groups of B cells and T cells within them. Okay, and all of them are going to be stationed there so that they can look at what types of foreign antigens are coming into the body. Okay, so what happens when they encounter a foreign antigen? Okay, so here we have, in this illustration, 3 different B lymphocytes. And again, we can tell that they're B lymphocytes or B cells because we have those Y-shaped receptors. Okay, and remember that the receptor of a B cell is just an antibody that's bound to the surface. Okay, so we have the foreign antigen represented by these yellow triangles. Okay, and the foreign antigen is going to be recognized by one of these B cells. Okay, and so the only B cell that has receptors that match up with that foreign antigen is letter C here, B cell C. Okay, so we can see we have that kind of triangular shape to the specific area of the receptor that would allow it to interact with the foreign antigen. Okay, so this B cell is going to be activated. These two are not. Okay, they're just going to remain inactive, or we can think of it as dormant or sleeping, however you want to think about it. We're just waiting for a foreign antigen to come that would match up with their receptors. Okay, so what's going to happen to this B cell? Okay, well, the first thing it's going to do is it's going to use receptor-mediated endocytosis to bring this receptor and the bound antigen into itself. Okay, after it's brought that antigen in, it's going to start processing it, and that's going to activate this cell. Okay, and once the cell is activated, it's going to start dividing. Okay, and it's going to form a group of B cells, activated B cells, that all have the same receptor on their surface. And that receptor is going to be identical to the receptor on the original activated cell. Okay, so it will have the same binding characteristics as the receptor on the original cell. Okay, and this B cell will also start producing a small amount of antibody at this time. Okay, this group of identical B cells that's produced from the activated cell is referred to as a clone. Okay, now if this is the first time this B cell has been activated, if it had just come out of the bone marrow and moved into a secondary lymphoid organ, it had never been activated before, it would be experiencing its primary response. Okay, so the primary response, since it's the first time this B cell has ever done this process, is going to produce a relatively small clone compared to the other type of response. And then those clone cells will differentiate or change. Most of them will change into these cells, which are plasma cells. Okay, now this, you can see the round nucleus in there, You can see how each cell is kind of oval shaped. You can see there's something kind of squiggly inside the cytoplasm there. That material that looks squiggly in there is representing rough ER. Okay, so it's telling us in the picture that this cell produces a lot of protein. Okay, so it will have a lot of organelles in it that help it with protein synthesis. So rough ER, ribosomes, Golgi apparatus. Okay, lots of all of those in there. Okay, and the protein that these cells are producing are antibodies. Okay, so these cells, these plasma cells, are basically like antibody factories. Okay, so these plasma cells will start making antibody. Since there aren't that many of them, they'll make a moderate amount of antibody. Okay, and that antibody will be around for a number of days, but eventually it'll start tapering off and go away. Okay, and these plasma cells will die off. Now at the same time, our clone cells, some of them will differentiate into memory cells. Now the memory cells are going to wait in the body for the next time the same foreign antigen appears. Okay, and when that foreign antigen reappears in the environment, the memory cell will become activated, and since it's been through this experience previously, it's going to have a much more robust response. Okay, so we're going to see again the receptor-mediated endocytosis, the formation of the clone, the differentiation of the clone cells, but the number of clone cells is going to be much higher, so the number of plasma cells that we produce is going to be much higher. The amount of antibody that's produced is going to be much larger compared to the primary response, and the length of time that these plasma cells are going to survive and produce antibody is going to be extended. Okay, again, when we make our clone, some of the clone cells will differentiate into memory B cells. So if we have this antigen appear again, we'll repeat this part of the process. Okay, so you only ever have one primary response, but you can have many, many secondary responses over the course of the lifetime of the organism. Okay, and how does that help us as far as different types of pathogens? Well, you can see that it doesn't matter whether it's a virus, a bacterial toxin, or a bacteria. There are B cells in your body that can deal with each of those. So they're giving us a great deal of protection from not only the pathogens, but the products of the pathogens, like the toxin. Okay, and antibodies have a number of different effects. This effect that we're seeing here is precipitation, where you take a small soluble antigen, link it together with antibodies, and turn it into something that can't stay in solution anymore. Okay, so something that's in solution can't be phagocytosed. Something that you make big and chunky that can't stay in solution, a phagocyte can gobble that up. Okay, you can also coat the surface of a pathogen with antibody, and this is what we refer to as neutralization. Okay, so if this virus is coated with antibody particles, it can't bind to a cell and infect it. Okay, so that protects us. We can also use antibodies to activate complement. Okay, so here are a couple of antibodies that have bound to antigens on the surface of a pathogen. The complement, when it sees those bound antigens or antigen-antibody complexes, will become activated, insert those pores into the membrane, and cause the lysis of whatever the antibodies are attached to. And we can also link together antibodies on cellular entities. Okay, so we have